lapel.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
So today we're diving into the world of plant-based compounds. We're focusing on one specific molecule. It's called 5-O-methyl-11-O-acetylhockin.
Or MAA for short, thankfully.
Yeah, MAA. Much easier.
And we've been looking at this research article. It uses some really interesting computational methods, basically computer modeling, to look at the potential of this MAA. It originally comes from the roots of a plant, Alkanna cappadocica.
Okay, so our mission really is to understand what these computer predictions tell us about MAA. Could it be useful in drug discovery? And maybe more importantly for a lot of you listening, how do these kinds of computer analyses apply to other natural compounds from plants?
That's the key connection, I think. These methods aren't just for MAA.
Definitely. Okay, so let's get into it. What is MAA fundamentally?
Okay, so chemically speaking, it's a naphthoquinone. It's just a type of molecule with a specific structure, two ketone groups, if you want the detail.
Right.
More specifically, it's a hydroxynaphthoquinone. And what's really interesting, like you mentioned, is it's not alone. It's related to other compounds, maybe ones you've heard of, like alkannin and chiconin. They're often used as natural pigments.
Ah, okay. Yeah, I think I've seen those names before, maybe in traditional medicine contexts.
Exactly. Those related compounds, they're found in the roots of a whole bunch of plants in the Boraginaceae family. So not just Alkanna, but also things like Lithospermum, Echium, Anosma, and Schusa, Sonoglossum too.
Wow. So it's quite widespread then, this type of compound.
It is, and that's a really important point if you're interested in natural products generally. It suggests that this basic chemical structure, might pop up in different plants and could have interesting biological effects worth investigating more broadly.
Right, so it's not just a one-off discovery in a single rare plant.
Yeah.
That makes sense. And didn't the paper mention some previous findings on MAA itself? Like before this computer stuff?
Yes, exactly. And this is where it starts to get quite compelling. Earlier research apparently showed that MAA they use the phrase impressive cytotoxicity against various cancer cell lines.
Impressive cytotoxicity, meaning it's pretty good at killing cancer cells in lab tests.
That's the gist of it, yeah. So that earlier finding is a big clue, right? It points towards potential therapeutic value, maybe as an anti-cancer agent, and it really highlights how compounds from plants can be, really valuable starting points.
Okay, so a natural compound, maybe anti-cancer activity. Sounds promising. But this study we're focusing on, it's all computational. Virtual analysis.
That's right. They used computational pharmacology. It's a field it's growing really fast. It uses software, databases, algorithms, basically using computing power to analyze biological data and predict how molecules might behave.
Like virtual experiments before you even hit the lab bench.
Pretty much. Saves a lot of time and resources, potentially. And a key thing they looked at is ADME.
ADME.
Yeah, it stands for absorption, distribution, metabolism, and excretion. These are absolutely critical in early drug development. Can the body actually absorb the compound? Where does it go? How does it get broken down? And how does it leave the body?
Makes sense. If it fails on one of those, it's not much use as a drug, is it?
Not really. So predicting these early is super helpful.
So how do they predict MAA's ADME stuff using computers?
They used a web tool. It's actually freely available online called SwissADME.
SwissADME.
Yeah. And it uses various models to predict all these properties just based on the molecule's chemical structure. And this is huge because you or anyone could, in theory, plug in the structure of another natural compound you're interested in and get a first look at its potential ADME properties.
That's fantastic. Really practical tool for anyone exploring natural products. So what did SwissADME suggest about MAA?
Well, one neat output is something called the bioavailability radar. It's a visual chart. It looks at things like the molecule's flexibility, lipophilicity, that's how much it likes fatty, versus watery environments, its size, polarity, solubility, a few key factors. And for MAA, the radar showed it sitting nicely within the optimal zone. Meaning the models predict it should have good oral bioavailability. So if you took it as a pill, there's a good chance your body would absorb it well.
That's a big plus for development, obviously.
Definitely. Then they used another model. This one's called the boiled egg model.
The boiled egg model?
Well,
seriously?
Yeah, it sounds funny, but it's quite intuitive visually. It predicts two things at once: how well a compound might be absorbed in your gut, and whether it's likely to cross the blood-brain barrier, get into your brain.
Okay, intriguing. How does the egg work?
So imagine a boiled egg diagram. The white part represents good predicted gut absorption. The yolk represents good predicted brain penetration. MAA landed firmly in the white.
Good gut absorption.
Exactly. But it stayed out of the yolk.
Meaning it probably doesn't get into the brain easily?
That's the prediction. Which could be good or bad depending on what you want the drug to do, right? But it suggests it might work systemically without causing, say, drowsiness or other central nervous system side effects. It's a specific prediction that helps guide the next steps.
That is pretty specific. Okay, good absorption maybe stays out of the brain. What about just general drug-likeness? Does it look like something that could be a drug?
Right, so that's where rules like Lipinski's Rule of 5 come in. It's a classic set of guidelines, sort of rules of thumb, in drug discovery.
Well, I've heard of that one.
Yeah, it looks at simple properties, molecular weight, hydrogen bond donors and acceptors, lipophilicity again. Basically, properties that tend to correlate with a compound being successfully absorbed when taken orally.
And MAA.
It passed with flying colors, no violations of Lipinski's rules. The numbers were all good: molecular weight 344.36, only one H bond donor, TPSA 89.9, log P 2.8, 6 H bond acceptors, all well within the preferred ranges.
So it ticks the basic boxes for an oral drug candidate.
It does. And it wasn't just Lipinski, it also passed several other sets of drug-likeness rules. Mueg, Gauss, Weber, Egan.
Okay, so multiple checks suggesting it has the right fundamental properties.
Exactly. And again, these are the exact same criteria you'd use to evaluate any natural product you were hoping could become a medicine. It provides a standard framework.
That makes sense. What else did they look at?
They looked a bit closer at lipophilicity. That log P of 2.80 suggests it's moderately lipophilic, which is often a sweet spot. And critically, it's predicted to have good water solubility.
Ah, solubility. That's important, right? Needs to dissolve to get absorbed.
Absolutely essential. If it can't dissolve in your body fluids, it's not going anywhere. Good solubility is a really desirable feature, especially for plant-derived things people might want to take internally.
Okay. And pharmacokinetics, how the body handles it.
Right, so digging deeper. High GI absorption predicted, which matches the boiled egg thing. Importantly, it's not predicted to be a substrate for something called P-glycoprotein.
P-glycoprotein.
It's like a bouncer protein on cells. It actively pumps certain molecules out of the cell. If a drug gets pumped out too efficiently, it can't work properly. So the fact MAA isn't predicted to be a substrate is good news. It might stick around inside cells better.
Okay, avoids getting kicked out.
Yeah.
Yeah. But then there's the CYP enzyme part. These are major enzymes in your liver that metabolize drugs. The prediction is that MAA might inhibit several of them, specifically CYP 1A2, 2C19, 2C9, and 3A4, but not 2D6.
Inhibit. So slow them down. What does that mean practically?
It means if you took MAA alongside another drug that's normally broken down by one of those enzymes, MAA could slow down that breakdown. That could cause the other drug's levels to build up in your body, potentially leading to side effects.
So potential drug interactions. That's a really crucial thing to know, especially for natural products people might take alongside conventional medicines.
Absolutely vital. It's a standard safety check.
Yeah.
And finally, they calculated a bioavailability score of 0.55 or 55%.
Which is good. Yeah, that's considered pretty good and suggests fairly consistent bioavailability, backing up the earlier absorption predictions.
Okay, so MAA is looking quite promising on the ADME and drug-likeness front. Now, the paper also mentioned medicinal chemistry aspects. What's that about?
This is about looking closely at the molecule structure for any potential troublemaker groups, features that might cause problems later. They looked for PAINs.
PAINs. Sounds bad.
It stands for pan-assay interference compounds. These are structures known to sometimes mess up lab tests, giving false positive results in drug screening assays. It's something chemists always look out for. MAA triggered one PAINs alert.
Just one. What was it?
It was for the quinone group in its structure, specifically quinone A.
And is that quinone group a major concern?
Well, it's something to be aware of. Quinone structures can be reactive. They've been linked to things like DNA strand breaks. Now, that reactivity might actually be related to why MAA shows cytotoxicity against cancer cells.
Ah, so the potential problem might also be part of its potential benefit.
Exactly. It's complicated, but it's a flag. It means you need to investigate carefully its mechanism and potential off-target effects. And finding these alerts, like this quinone, is super relevant for evaluating other natural products too, as similar structures exist in many plant compounds. Identifying them early helps.
Right. Forewarned is forearmed. Any other structural alerts?
Yes, one Brink structural alert. It's for an isolated alkene group. Again, it just flags a part of the molecule that might be chemically reactive or unstable.
So maybe something that would need to be chemically modified if this were developed further.
Potentially, yeah. It highlights that often the natural compound itself is just the starting point. Medicinal chemists might tweak the structure to improve stability or reduce unwanted reactivity, optimize it.
Makes sense. Refine the raw material.
Precisely. But on the plus side, MAA was predicted to have lead-likeness.
Lead-likeness?
Yeah, meaning it has the kind of properties that make it a good lead compound, a promising starting point for a drug discovery program. It's got potential.
Okay. And could we actually make it? Or do we have to rely on extracting it from the plant?
Synthetic accessibility. Always the practical question. They used SwissADME to predict that too. It got a score of 3.94.
Is 3.94 good or bad?
It's pretty good. The scale kind of goes from 1, very easy, to 10, very difficult. So 3.94 suggests it should be relatively straightforward to synthesize in a lab.
That's a huge advantage. If it looks promising, we can actually make more of it reliably.
Exactly. Relying solely on plant extraction can be limiting. Being able to synthesize it is key for development. This applies to any natural product candidate, really.
Okay. Now, the study didn't just stop at predicting properties. It also did molecular docking. What was that for?
Right. Molecular docking is like a virtual simulation. It tries to predict how a small molecule, like MAA, might fit into and interact with a specific target protein in the body, like a key fitting into a lock.
And what proteins did they test MAA against?
They chose 2 important proteins involved in inflammation, TNF-alpha and NF-kappa B.
Inflammation targets. That's relevant to a lot of diseases.
Definitely. And many plant-based remedies are traditionally used for inflammatory conditions, so it's a logical place to look.
So what did the docking show for TNF-alpha?
It predicted a pretty stable complex formed between MAA and TNF-alpha. They even pinpointed specific interactions, a hydrogen bond with an amino acid called leucine 233, and something called a pi-pi interaction with tyrosine 135.
Okay, specific connection.
Yeah, and the docking score was good, minus 6.964 kilojoules per mole. Generally, a more negative score suggests tighter binding. So this hints that MAA might interact well with TNF-alpha.
Suggesting potential anti-inflammatory activity via that pathway.
That's the implication, yes. It's a plausible mechanism based on the simulation.
Interesting, so maybe anti-cancer and anti-inflammatory. What about the other one? NF-kappa B.
The interaction there was predicted to be less stable. They saw some potential hydrogen bonds, but the overall docking score was much weaker, only minus 1.567.
Okay, so not as good a fit.
Doesn't seem like it, based on the simulation. So the researchers concluded that the interaction with TNF-alpha looked more promising, suggesting that might be the more relevant target if MAA does have anti-inflammatory effects.
Got it. So the computer models provide clues about how it might work, not just if it gets absorbed.
Exactly. And this, I think, brings us back to the bigger picture for you, the listener, especially if you're interested in other plant compounds.
How so?
Well, these methods they used for MAA, SwissADME, for the ADME, and drug likeness predictions, molecular docking to look at protein interactions, these aren't specific to MAA at all. They can be applied to study any compound, including thousands of others derived from plants.
Right, so this study is like a case study for a much broader approach.
Absolutely. The same ADME criteria, the same drug likeness rules, the same docking techniques, they're all tools in the toolbox for evaluating the potential of any natural product. SwissADME, being free and online, is particularly powerful for getting that initial screen done quickly and cheaply.
It must speed things up massively compared to doing everything in the wet lab from the start.
Hugely. It lets researchers prioritize which compounds are most likely to be worth the time and expense of further lab work. You can screen hundreds or thousands of structures computationally first.
That's definitely a game changer.
And remember the pains and Brink alerts, those structural flags.
Yeah, the quinone and the alkene.
Identifying those computationally is another example of how these tools help. You can spot potential liabilities, toxicity risks, stability issues early on for any natural product structure you analyze. It helps manage expectations and guide research.
Like a computational heads up for potential problems down the road.
Exactly. It allows for a more informed, efficient approach to exploring the incredible chemical diversity that plants offer. It's about using computers to help us navigate that vast natural pharmacy more effectively.
This has been really illuminating, a great look at MAA, but also at how these computational tools are transforming natural product research. So, summing it all up, what are the really key takeaways?
Well, for MAA specifically, the in silico or computational analysis suggests it has pretty promising properties. Good predicted absorption and bioavailability if taken orally, it meets key drug likeness criteria, and it seems relatively easy to synthesize. Plus, the docking suggests it might have anti-inflammatory effects by interacting with TNF-alpha.
But crucially, these are still computer predictions, right?
Absolutely critical point. These are predictions. They provide a really strong rationale and a great starting point for doing the actual lab work the experiments needed to confirm these predictions and really explore MAA's therapeutic potential.
Right, so it points the way for future experiments. And thinking more broadly, given these promising results for MAA, and knowing these powerful computational methods can be applied across the board to countless other plant compounds, well, it really makes you think, doesn't it?
Yeah.
What other amazing natural compounds are out there in plants, just waiting, holding undiscovered potential that maybe, just maybe, these kinds of computational explorations can help us finally unlock?
It's a huge frontier. The plant kingdom is an incredible source of chemical diversity, and these tools are helping us explore in ways we couldn't before.
A fascinating intersection of nature and technology, potentially leading to new medicines. Really makes you wonder.
Thanks for tuning in to Bloom's Cast, brought to you by blooms.io. Stay curious, stay connected, and we'll see you next time on the frontier of natural product innovation.
Pharma